I first invite Dr. Timothy Lai. He's going to speak for Time for Change, integrating faricimab into first-line therapy for AMD. Dr. Timothy Lai is currently coming from Department of Ophthalmology and Visual Sciences at Chinese University of Hong Kong. Professor Lai is honorary clinical professor at Department of Ophthalmology and Visual Science. He is director of Retina and Macula Center at Hong Kong, has published over more than 260 international peer-reviewed papers, and has been awarded with Nakajima Award at APAO and Senior Achievement Award at APAO and the Constable Lecture Award at Asia Pacific VR Society and Senior Achievement Award from APAO. Professor Lai serves on the editorial board of over 10 international ophthalmic journals. His other leadership roles include Vice President of APVRS and Chair of our Global One Advisory Board. Professor Lai clinical research interest lies in medical and surgical management of retinal diseases, particularly PCV, CSR, myopic retinopathy, and CNVM. May I request Professor Lai to give his presentation.
Thank you very much, chairs, and thank you, Roach, for sponsoring this symposium. So in this first part of the symposium, I'm going to talk about neovascular AMD, how faricimab can be introduced as a first-line therapy for treatment of neovascular AMD patients. So these are just some disclaimers. Let's look at the landscape so far. If you look at neovascular AMD, the incidence, it's actually expected to increase a lot in the next 20 years or so because of the aging population. And there's definitely a lot of unmet needs there. So patients frequently need frequent injections. This will lead to patient and caregiver burden. Patients might not go back to follow-up visits, and we therefore get suboptimal long-term outcomes because of insufficient treatment. So faricimab is a bispecific antibody which can target both Ang2 and VEGF-A. So you can see this molecule, one arm of the FAB blocks Ang2, which can stabilize the vessel, reduce vascular leakage, reduce inflammation, and the other portion blocks VEGF-A, which inhibits neovascularization as well as reduced vascular leakage. It also has a modified Fc portion, which can reduce the inflammatory potential. Faricimab has undergone a lot of extensive and ongoing clinical trials. And you can see in DME, we have our Yosemite and Rhine, and then a long-term RONEX study. Neovascular AMD, we have TANEL, Lucerne, Avenel X, as well as the PCV sub-study, which is called the CELWIN. In RVO, we have Barleton and Camino for both branch and central retinal vein occlusion. And we also have myopic CNV, which is currently having the Po-Yang trial, which is still undergoing. So if you look at the long-term outcome, we actually have more than 1,000 patients in the long-term ALPHA-NEX study, which the results will be reported later this year. If you look at the real-world experience as well, we have very good both Roche-sponsored study as well as independent studies with over 150,000 patients having injected with faricimab. So why is dual inhibition important? So let's go back to the basic pathophysiology of Ang2. So in healthy tissue, you get a low expression of Ang2, and this can actually promote vascular stability in these healthy tissue. But when you get various insults, for example, with oxidative stress, you get an angiogenic switch, and this will increase Ang2, which will displace the Ang1 from the Tie2 receptors, leading to various pathologic states, including leukocyte transmigration, vascular leakage, and upregulation of cytokine, making it more sensitive to VEGF. In the clinical studies, we actually showed multiple biomarkers that dual inhibition is very useful compared with VEGF inhibition alone. So, for example, if you look at the neovascularization and vascular leakage markers, with the use of faricimab, you get greater reduction in fluid and OCT. get greater resolution of intraretinal fluid as well as greater resolution in PED. In terms of leakage, you also have very good evidence from DMO and RVO study. You can get better reduction in macular leakage and heart exudates with dual inhibition compared with inhibiting VEGF-A alone. For the Ang2 side, you also get reduced inflammation as shown in the DME study with reduced hyperrefractive foci and also reduced development of ERM in DME cases. Faricimab actually suppresses Ang2, which aflibercept does not. So if you look at the aqueous samples study from the phase 2 trial as well as from TANA-Lucerne, one dose of faricimab, you can actually get absolute reduction in the free Ang2 inside the eye, and this persisted for more than 8 weeks. For aflibercept, you can see there was actually no significant change in the aqueous humor level of Ang2. People might be asking whether we can increase the dose of the anti-VEGF injection to get further improvement in terms of the anatomical outcome. But if you look at the Harbor and Pulsar study, so Harbor increased the dose of 0.5 milligrams of ranibizumab to 2 milligrams, and in Pulsar it increased from 2 milligrams to 8 milligrams of aflibercept. But if you look at the mean OCT change, basically these curves overlap each other, so did not show any further improvement. And in the document published by the FDA, when the aflibercept 8 milligrams was approved for neovascular AMD, FDA stated that the study did not establish consistent treatment benefit of high dose of aflibercept in retinal defect drying, and the clinical effect of high dose of aflibercept is questionable. So how does drying we can achieve the disease control? So if you look at our registration trial, the TANA-Lucerne study, these patients initially they would be dosed at a 4-weekly interval for 4 doses, and then if you look at this head-to-head comparison over this time, you can see comparing with aflibercept at this head-to-head level, we can get further reduction in central retinal thickness as well as a higher proportion of patients with absence of subretinal and intraretinal fluid by about 10% during this head-to-head period. So it showed greater anatomical improvement by having dual inhibition of using faricimab. Another way to look at this is that we can actually achieve faster drying of the macula as well. So this is the Kaplan-Meier survival curve showing the first absence of intraretinal and subretinal fluid with faricimab versus aflibercept. And you can see you can achieve this 75th percentile with the only 2 injections of faricimab compared with a median of 3 injections for aflibercept. So you can get this about 4 weeks earlier. Another very good marker, as you know, PEDs are very difficult to treat, and you can also get very good reduction in the PED height in the head-to-head period. You can see additional 17 microns reduction. So now we have the 8 milligrams of aflibercept available, but unfortunately we do not have a head-to-head study comparing faricimab versus aflibercept. So we can do some direct comparison between faricimab and aflibercept in the TANA-Lucerne study, also a direct comparison between aflibercept 2 and 8 milligrams in the Pulsar study. But how do we do the comparison between faricimab and 8 milligrams of aflibercept? So we can do something called matched adjusted network meta-analysis. So by having this in direct comparison. So if you look at these, they actually handpicked all the matching the subjects in the 2 study, and then they found that by having this comparison, it showed faricimab seems to be able to dry the macula better compared with aflibercept 8 milligrams. So what's the importance of having the drying of the macula earlier? So in the TANA-Lucerne study, it's been shown that if you can get subretinal fluid and intraretinal fluid resolution early within the first 12 weeks, you are much likely to have extended treatment interval later on. So this is more than 80% chance of having the patients in the 16-weekly dosing compared with the 8-weekly dosing. So it is associated with extended durability. In the Retina and AMD study based on the IRIS registry, it showed that with the use of faricimab, you can get very rapid improvement in vision, as well as reduction in CST, as well as improvement in vision. And this is actually reflected by a low number of injections as well. And therefore, it showed that the clinical trial data is very well reflected in the real world. Another real-world study is the ChANGe study, also showed that in this patient, they had a substantial reduction in intraretinal fluid and subretinal fluid after being put on faricimab treatment, and the number of injection is also very low. In terms of PED, also 2 real-world studies show that by having faricimab injection, you can get rid of these very difficult to treat PED very quickly. Of course, in Asia, PCV is very important, and that's why we performed the SALWIN study, which is a phase 3b/4 study, multi-standard study, including sites here in India, to look at the use of faricimab for PCV patients. So patients, the study design is quite similar to Tana-Lucerne. Patient had initial dosing in the 4 loading doses, and then had disease activity assessment, and then put on either 8, 12, or 16 weekly dosing. But in the second year, it's a bit different. So we can actually have a treat and extend period up to 20 weeks. So this is the only study which allowed up to 20 weekly dosing. So here is the interim analysis at week 16. You can see patients improved by 7.8 letters with faricimab treatment in these PCV cases, and very good reduction in macular thickness. So it's very consistent with what we saw in the Tana-Lucerne study. Most remarkably is the absence of subretinal and intraretinal fluid with treatment. So we can achieve 80% at the week 16 mark. And also for those patients with ICGA confirmed polypoidal lesions, 51% complete regression rate was observed, and this is very encouraging because previous monotherapy study using anti-VEGF were not able to show this kind of complete regression rate. So just some take-home messages. So faster drying and greater anatomical improvement with faricimab versus aflibercept has been shown in the Tana-Lucerne study. Drying and anatomical improvement data is also reflected in the real world, and rapid drying has led to potential to have extended durability. So now I'll talk about how durability can reduce the treatment burden. So if you look at the patients in the studies, you can see that in the first year, 78% of patients could be dosed every 12 or 16 weekly. And this was actually maintained in the second year. Again, about 78% could be maintained on Q12 weekly or Q16 weekly dosing. If you look at the swim lanes in these individual patients, a lot of patients could actually be put on the Q16 weekly or Q12 weekly dosing without dropping down at all. And for these patients who were always on Q16 weekly dosing, they actually achieved very stable best corrected vision gain and CST reduction throughout the 2 years. You can see they did very well despite just being dosed a few times in the second year. So one of the ways to look at how these patients do in the long term, we can actually, because the Tana-Lucerne didn't have allow further extension beyond 16 weekly dosing. So we can potentially look at the criteria if these patients who were already on 16 weekly dosing, if they could actually be potentially further extended. And you can see about 56% of patients could potentially have further extension from 16 week to 20 weekly dosing. In the faricimab AMD trial, basically, we use a disease criteria which is quite reflective of the clinical practice. So you can see these patients in terms of dosing, Q12 weekly dosing or more could be achieved in 78%. But if you look at another criteria, for example, looking at both vision and anatomical criteria, which was actually used in what's in the PULSAR study, they looked at 15 letter loss and CST increase of 250 micron or new macular hemorrhage, then you can see actually this rate durability can be extended in about 96% with the Q12 weekly dosing or more. So definition of active disease in clinical trial is actually quite important. So in the TENA-LUCENT trial, we use vision but if you have this hypothetical disease activity criteria using the vision and anatomic, you can get this to 96%. So if you look at aflibercept 8 milligrams trials, Candela, they actually use which is about 53% could get up to Q12 weekly dosing. PULSAR, they use vision and they get up to about 83%. So it's important to look at the extent criteria when you are interpreting these clinical trial data. Looking at the real-world studies, for example, for retina AMD, far-wide AMD, you can see in the second 6 months of the study, very consistent with the real-world, with the clinical trial data, only about 2.4 injections. So few injections in the second year show that durability exists in the real world. So you can see real-world data reflects the clinical trial data, and based on other independent real-world study, both in Morpheus and Japan, again, over 70% in the Morpheus study could be dosed 8 weekly or less frequent than 8 weekly, and in Japan, again, 67% can be dosed 8 weekly and less frequently. So just take-home message about durability. Faricimab showed that extended durability can be shown in the TENA-LUCENT study, and that's also reflected in the real world, which can help our capacity issue in the real world and also reduce the treatment burden of our patients. So finally, just some cases. This case, treatment-naive neovascular AMD, the left eye already and only eye, right eye is poor vision because of a corneal scar, so it's a left-only eye patient. So the patient asked me what treatment should I get. She wanted the best treatment. So treatment-naive patient. So this patient, we offered her having faricimab injection. You can see a lot of shrimp and also PED, so quite a difficult to treat patients if you see this kind of lesion characteristics. So given first dose of faricimab, did very well already, reduction of PED, substantial reduction of subretinal fluid, second dose was given, and so these patients 4 loading doses and then injection period, so we could go up to 16 weekly dosing straight away after 4 loading doses, and the patient is quite happy with the 20/50 patient. And because this patient is an only eye patient, I'll just keep on having the Q16 injection at the moment. So no safety signal was encountered in this case as well. So another case, this is a treatment-naive PCV patient which was actually enrolled in the SALWIN study. Left eye already or PCV scar and a vision of 320, and this eye 20/40 vision to start with. You can see a baseline FA and ICJ showing the polypoidal lesions very well. So patient put on faricimab, and you can see after 1 dose, flattening of the PED as well as resolution of subretinal fluid with good maintenance in vision and some improvement in vision later on. So this patient, after 4 loading doses, extended to the Q16 weekly dosing. And at week 16, we performed ICGA. You can see very good complete regression of the polypoidal lesion. So this patient maintained our Q16 weekly dosing, and after 1 year, he was able to be further extended to Q20 weekly dosing, and this is after the 7th injection been given. So very stable disease activity at the moment. 4 loading dose followed by 16 weekly and then Q20 weekly dosing. And again, no safety signal was encountered. So just the key takeaways now. So we have demonstrated why faricimab could be a very good first-line treatment to optimize your patient's outcome. How is this achieved? We can achieve this by having better drying, very good durability and also by dual inhibitory pathway of both Ang2 and VEGF, so the 3Ds. And how do we know this? Of course, we've got very good clinical trial data to support, as well as our real-world evidence to support these 3Ds in achieving the effects. So with that, thank you very much.